Thanks. Yeah, thanks for the opportunity. I appreciate the chance to be here tonight to share what we're doing. So I guess I would start off by emphasizing this is really a team effort and community-based research. This is a big problem, as you heard. And so it's something, it's you can see a partial list of people below here, the Aroostook Band of the Micmac Nation, which is a tribal community in Maine we work with, Upland Grassroots, which is a community advocacy group in Maine, corporate partners, CEM and SciAx, that are developing technologies that we've used to characterize PFAS, the Jefferson Trust, VIPC, USDA, and then most recently the EPA, who have been able to support a lot of the scientific research we're doing. And so Mike, who's in the audience here, is a colleague of mine at UVA that I work closely with on this as well. So, it really is a team effort. And so I'd say it's universities, academic partners, industry partners, corporate partners, all working together to try to solve these problems. And so some of the things I'll talk about at the end were solutions that we're trying to come up with. One is detection tools. So if you're a grower and you want to know whether you have PFAS on your farm, Mike and I are working towards developing tools that would allow you to test that at a low cost. Mike and I are also working on trying to bring deliverables to growers. So for example, if you know you have PFAS in your soil or you've applied biosolids that contain PFAS, what information can we give you as a grower to help you mitigate that or make best decisions as you're harvesting your crops? And then, in particular with the Micmac Nation, there's a lot of culturally important plants that aren't cultivated plants, but are very important. And so, studying how ecosystems and food webs broadly are impacted, not just sort of agricultural crops. So I'll kind of touch on all those things tonight. And please make this interactive. Interrupt me if you have questions along the way. I've tried to gather a lot of information, to kind of provide information too. So, please feel free to interrupt. So, first, I wanted to just explain for those. I think for this audience, most of you are familiar with what PFAS is, but just to give a little bit of background and history on what it is. This is a little bit of a history or timeline of what pair and perfluoroalkyl substances, or PFAS, came from. This was actually invented in 1938. It was actually invented in part to mitigate fires or other sort of issues surrounding the generation of nuclear energy. It was sort of discovered at that time, and then interestingly was found to be very useful as a non-stick cookware surface. So in the 1940, production began. Scotchgard was, in the 1950s, one of the first commercial products that kind of hit widespread use. Teflon in the 1960s, there's sort of this long history of sort of commercial products that were developed. And then 80s kind of went global, and it really wasn't until the early 2000s that people began to appreciate or kind of began to see some of the adverse health impacts associated with PFAS. And so, kind of mid to early 2000s, EPA began to regulate and then ultimately sort of phase these chemicals out. And so, here we are today with these action plans, and we have regulations for drinking water, and I'll show you those, but biosolids and other categories of materials, there's draft guidance, but no strict regulations on PFAS limits in these materials. So this is kind of an ever-evolving timeline. So we're kind of in that regulatory question mark right now. Yeah?
Are PFAS still entering the, water cycle as quickly as they were? Or is it, how is the level of it over time? Is it increasing, decreasing?
I think it's hard to say. I think people, a big part of it is actually just testing, honestly. To be able to test and track its movement is very challenging. So for that reason, it's maybe hard to say with certainty where or how. So I think we're kind of at a point where we're trying to figure that out, actually. And agriculture and biosolids being one source of material where it can bioaccumulate. So I think maybe a way to answer that is people are trying to figure out now where pinch points are, or accumulation points, or kind of major point sources that could be generation sources downstream in water, and biosolids being one of them. So, so I think they became very popular because of their flame retardant and their water repellent properties. So, firefighting foams, food packaging, water resistant clothing, paints, cosmetics, pesticides, you name it, right? They're in all these different products. And so it's that water repellent nature that makes them very popular, as well as the fact that they're used in aqueous firefighting foam. So those are kind of the two of the major sources that really kind of turbocharge their production. But there's a lot of others too. And so, one question is, why are they forever, or what makes them last forever? So, because another name for these are forever chemicals. You'll see that in the news. So, so up on the right there, two structures, PFOS and PFOA. Those are the two most abundant, kind of breakdown products, or most persistent ones that show up in the environment. There's a little bit of chemistry for those of you that like chemistry. So there's a periodic table. This is a periodic table of the elements based on their electronegativity. So electronegativity is how strong an atom attracts an electron in a chemical bond. So if there's any organic chemists in the room, or people that had a chemistry background, you'd know that the carbon-fluorine bond is the strongest bond in organic chemistry, right? So it's the most electronegative element, fluorine, which really tightly bonds itself to that carbon, and that's what makes that bond so strong. And in essence makes it last forever. So another interesting thing to point out, and Mike, you can correct me if I'm wrong since you're a biologist, fluorine is not an essential element for life. It is an element, but there's no known terrestrial process or no biological role that's essential that requires fluorine, right? And so that's one reason why it's very unlike other types of contaminants where biotransformation occurs in the environment, because these elements are involved in some way in biological processes. That's not the case with fluorine. So that's another reason why they last forever.
Can you spell fluorine? It's so much easier for me to remember.
Yeah, it's Fluorine. Yeah, I have it here. Yeah. Or I can send it to you.
It's like chlorine.
Yeah, I guess it's similar spelling to chlorine. Yeah, CHLOR. So it's one of the elements, but not one that's essential for life. So there's over 4,000 known compounds that were classified as PFAS. So different. I just showed you two that were very common, but there's many more. It's estimated that they take over 1,000 years to biodegrade in the environment. Just based on what's known today. And that's obviously projection, but they last forever, right? And that's sort of what we expect. And they're linked to a wide range of health issues. So that's another graphic that kind of shows you different products that are made using these compounds. And on the left is actually from the European EPA, but the US EPA has a similar graphic. And this I show because it illustrates diseases or other sort of health conditions where there's partial or direct evidence to PFAS exposure. So the ones I would point out are primarily immune suppression, so it's reduced response to vaccines and other antibody responses, low birth weight, mammary gland sort of reproductive development disorders, lung damage, kidney damage, thyroid disease. These are ones where there's enough association that EPA could make a moderate to strong association. There's a bunch of others that are implicated, but not enough evidence yet to implicate them strongly. So yeah, I could go through all those different examples, but I won't. One I would point out that I think is particularly noteworthy is the immunotoxicity of PFAS. And this relates to some work that Mike and I are starting to do with colleagues at UVA Health and other groups on ground. So these are some links, and I'm happy to share this afterwards so you can read all these links and read these articles if you're interested. So early studies indicate that circulating antibody levels for specific vaccines are lower in children who have elevated exposure of PFOS and PFOA at birth. And in some cases, the circulating levels were below the threshold necessary for efficacy. So what that means is that if you were exposed at birth because your mother's blood serum level had significant levels of PFOS and PFOA, 5 years down the line, on average, in this small study, your antibody levels were lower, right? So you had a lower immune response. And in some cases, low enough that it was as though you did not have the vaccine in the first place. And so PFAS has been detected in breast milk, so it's been shown that it can be transmitted from mother to child. And so children and other vulnerable populations have a lot of different exposure risks to PFAS, right? So at an early development stage, exposure to PFAS compounds like this can have adverse effects on your immune system development and all sorts of other issues. And so this article I show because it's one that was kind of striking. So this is from 2020. This is a study that showed carpets and paints in daycares in California. If you measure the circulating dust levels in these daycare facilities, you find that they have, significant levels of PFAS present in the carpet and then in the dust, right? And so children are much more exposed because, as you can see in the graphic, children crawl on the ground, they roll around on the carpet, they come into direct contact with it, right? So it's another contact source that was unappreciated that could lead to, greater exposure risk and ultimately immunotoxicity. There's also examples of this in agriculture. So this is a study from 2024. It was a 5-year cumulative study. It was 181 women in this test. So these were agricultural greenhouse workers, specifically female workers. So female are more at risk than men in some cases, just due to the preponderance of fatty tissues, say in the breast or the reproductive organs, that make them more susceptible. And so in the study, what they found was that the major PFAS in serum, PFOA, PFOS, was 50% higher than the general population when this was age adjusted. And so the outcome of this study, suggested that PFAS, pesticide exposure is another underappreciated risk factor to consider, right? So in other words, these pesticides are formulated with low levels of PFAS, and so if you work in a greenhouse or a confined space where you're applying these, you're inhaling this and you're at much greater risk of taking it up. Yeah?
So is that primarily from, do they see that it's primarily from pesticide exposure, or was that hydroponic, or how was that?
This study, I believe, was I'm not entirely sure whether it was a hydroponic or not. I don't think it was. I think this was this is in Denmark, and it was a cut flower greenhouse. So I think they were growing poinsettias and other flowers there. There's some other studies like that as well. I don't recall one. I don't know, Mike, do you, about hydroponics? I don't think I've seen a hydroponic specific one. Yeah, it doesn't mean that's not out there. I mean, I just missed it. But this one, I think, is because of the larger cohort. It's a 5-year cumulative study. It kind of shows consistently this outcome. So it's another risk factor. That's agriculture specific. So kind of before I jump into the numbers, I wanted to give a sense of scale for this. And so there's a lot of numbers you'll see as you read about this. There's a lot of different units. I'm an engineer, so I'm trying to organize all these units so we're all kind of talking the same language, right? So 1 part per million, that's 1 ppm. That's equivalent to a drop in a bucket. 1 part per billion is equivalent to a drop in a swimming pool. And if you look online or you kind of read about current EPA regulations for drinking water, usually you'll see a phrase to the effect of 1 drop per 5 Olympic-sized swimming pools, right? So that's that 1 part per trillion. And so a nanogram per mil is a part per billion, a mg per liter is a part per million, a part per billion is 1,000 parts per trillion, and a part per million is 1,000,000 parts per trillion. So I'll kind of, as I go through the data, you'll see, and I'll remind you, but this is just so that I've given you the units so you can follow along. So this is the final EPA drinking water regulations. This is just directly from the website. I cropped it. So PFOA, PFOS, maximum containment level is in the parts per trillion range. So that's the drop in a swimming pool, or 5 Olympic-sized swimming pools. And so some of the other ones are at 10 parts per trillion, right? So we're talking, exceedingly low levels that are allowed. And a lot of this is also just because that's as low as you can possibly go and still detect it, right? Doesn't necessarily mean that that's a safe threshold. And then this is from the Environmental Working Group. This is a map that's kind of continuously updated. You can go online and see it. I guess it was last updated August 14th. That shows drinking water where there are tests that have been reported that are above the proposed limit that I just showed you before. That's the light blue dots. The kind of the medium blue are below that. Military sites and other known sites are in purple and orange. Those are other sites where there's known to be high levels of firefighting foam, industrial chemicals, or other sources of PFAS. And those are in part to try and correlate spread relative to point sources, right? So you can see the eastern seaboard. Virginia's kind of in the middle there. You don't see a lot of dots. The Northeast, where I'm from, is covered in it, so I'm probably covered with it too. But But that gives you just a sense of how pervasive the problem is and how widespread it is.
But Brian,
Yeah.
we haven't done a whole lot of testing, have we? I mean, could it be?
Yes.
Yeah.
Yeah, so some of it is just, I think, a lack of testing probably as well. So, yeah, you can only report what you've tested, really. So that's probably part of it as well. But yeah, they have a good website where this is an interactive map, and you can kind of learn more about, each dot you can click on, learn about how it was collected and so forth. But yeah, I'd point out too, and I think this is kind of, to me, was kind of striking when I first learned it, that this was just drinking water. If you think about livestock, it's a very different story. So in this case, there aren't draft EPA guidance for this. So Maine has kind of led the way in most of these things, and so we work with colleagues in Maine. So I took this from the Maine website. So this is recommendations they give to cattle farmers and dairy farmers in Maine. And so there's some things that I think are important to point out. So in Maine, for example, action levels are 3.4 parts per billion for beef, and then 210 parts per trillion for milk, right? So parts per billion, that 3.4, that's 1,000 times higher than the drinking water limit. And 210 parts per trillion, that's hundreds of times greater than the drinking water limit. So that points out that these things bioaccumulate, right? So they bioaccumulate in us, they bioaccumulate in our food. And so they have a guidance level of 6 parts per billion. So if you have less than that, so again, that's 1,000 times greater than the drinking water limit. The land is considered adequate for producing forage. Forage is the main source that leads to this bioaccumulation in cattle. So they recommend soil testing as being most actionable for assessment, for your risk to livestock. And so, again, just to emphasize, that's 1,000 times greater than the drinking water limit. So that kind of speaks to this bioaccumulative nature of it. Yeah.
Is Maine subject to more biosolids being spread, or do we know this because of testing?
Yeah, I'll have some comments on that later, but Maine, I think Maine kind of leads the nation, I would say, in how we've learned about this, and that's through the unfortunate learnings in Maine that application of biosolids led to significant contamination of agricultural lands, and so because of that, they were one of the first states to institute a ban, and so I guess that's kind of why Maine and where this comes from. It's kind of based on what they've learned. Yeah.
Are the numbers similar, like for organics or grass-fed, or is it the same?
I don't know. I don't remember from this, the recommendations, whether it broke that out either way. So I'm not sure. Yeah.
Do you plan to address the difference between testing surface water versus testing for soil or biosolids itself? Because I'm confused by that.
Yeah, I have some comment, like some discussion of it later, but interrupt me if it's along the way.
Appreciate it.
And I guess this is to your question about Maine. So these are just 2 articles that were written. A lot of you may have read these articles. The one is in the New York Times, the Something's Poisoning America's Lands, and then the One State's War on Forever Chemicals in Milk talks about Maine specifically in New Republic. So I think Syngro, the same company you were talking about, this was back in 2022. They were testing at ranches in Texas. They're headquartered, I think, in Fort Worth in Texas. On these farms, they were finding hundreds, tens of thousands or hundreds of thousands of parts per trillion levels in animals, biosolids that were applied to the cattle farm and then to the drinking water, right? So that was one way in which biosolids began to be linked to some of these issues.
Can I just mention that all of these articles are available on Don't Spread on Me?
Oh, good. Okay, I didn't know. Well, yeah, I would. Yeah, that makes sense. And then, you'll see your ranches in Michigan have been shut down because of, after testing biosolids in meat where elevated levels were found. And then Maine in 2022 banned the use of biosolids based on, kind of what was happening and set up our limits for PFAS in meat and dairy. And that's what I showed you a moment ago. Yeah, Brian.
So the impact of closing down the spreading of biosolids in Maine meant that they were then exporting them out to New Hampshire and Vermont and New York, Pennsylvania. Anybody could take them.
Canada.
And it's the same And is Virginia going to do that?
You're talking about the
Yeah.
application or the ban?
Are we going to ban it in Virginia? Probably not.
I'm not sure. I would hope. I would hope this information is kind of informative, for people. But yeah, I don't know.
Take more from other states.
So yeah, now that this is sort of the linkage between biosolids, agriculture, and I'll kind of give more examples, but these are just some definitions to kind of explain what they are. So EPA's definition, sewage sludge is a precursor that's been treated to become biosolids. So I think the terms are used interchangeably, but that's kind of the EPA's definition. So there's two classes, Class A and Class B. Class A has strict requirements, mainly for pathogen levels, and so that's the millorganite you see there, that you can buy at Home Depot or your local store. Class B has less strict standards and restricted use, so you can't sell it bagged to the public, but those are ones that are primarily applied to agricultural lands, reclamation sites. So low levels of microbes and pathogens, but regulated levels in the sense that there's a low risk of spread to the environment. And so, yeah, that I took directly from the EPA's website. So this is kind of how biosolids are made. I think you explained at the beginning, Stuart. So it can come from your septic system, your house, industrial sites. It goes through a series of screening and sedimentation tanks. It then goes through a series of heating, oxidation, irradiation, other steps to sort of inactivate pathogens and break material down. That's then sent back to settling tanks. So if it, at that point, it's Class B. Otherwise, it goes through tertiary additional treatments, which disinfect, with chlorine or UV light to inactivate the residual pathogen. And that's the Class A that I mentioned before. So that's kind of a little bit of how this stuff is made. But biosolids being one, there's other ways in which this enters the food system, right? So I talked about the biosolids and gave those examples earlier. Whether it's irrigation water, whether it's biosolids, that enters soil, that then gets into crops, comes into direct contact with farm workers, animals consume forage or other grains or grasses, their poop goes back into the soil, which recycles it, or that ultimately ends up in the food we eat, right? So that's this kind of, PFAS moving through the agricultural landscape. So almost 30% of agrochemicals, such as pesticides, are fluorinated to increase stability. A lot of food packaging is also fluorinated to make it a reduced oxygen or moisture barrier. The process of fluorination, you can generate a small amount of PFAS as a byproduct, but as I mentioned before, it's parts per trillion, so it's an exceedingly low level. So even a small amount that's generated is still enough to be significant. And it's present in soil amendments such as biosolids. And so, that's agriculture, but then you kind of imagine it spreading through food webs as well. So I'm sorry the graphic's not clear, but you can kind of imagine that ending up in soil, foraging, animals, wildlife consume it, and it kind of begins to spread from there. And so this on the right is a graph from this is a study that was done in Michigan, looking at accumulation levels of PFAS in different fish types. Many of these are at levels above 10 parts per billion. So again, that's thousands of times greater than the drinking water limit. And so there's a lot of different studies that kind of show this to be the case. Like earlier when I was showing that graphic and kind of talking about there's a limited number, like the immunotoxicity, for example. I think there's really only 6 studies that were in that EPA report. There's a lot more animal studies to substantiate that.
Okay, that's fine.
Yeah, but I think with cancer, there's a lot of kind of in vitro studies, or but I don't know as much.
I don't mean specifically cancer, but I just mean
just in general.
Yeah.
I don't know if there's a linkage between a specific compound and disease, like one is worse than the other.
Concentration of it.
I think so that's probably, I think that goes back to the parts per trillion, right? The levels that the EPA set, based on risk. So I think that kind of was extracted from the studies, like the ones that I mentioned. So, but I don't think there's a definitive or clear answer about that. That's still an open question. Yeah.
Sorry, is there a tipping point or a certain event that led the EPA to start looking into this as a health detriment, or was it just something steadily that happened over time?
I think, like back at the timeline here, kind of the early 2000s, if you kind of go back to 3M or like sampling workers at facilities, they if you read about the history of like the 3M, for example, one of the toxicologists there was doing routine blood testing and finding compounds that they couldn't identify what they were, and so they were realizing that it was accumulating in people's bloodstream. And that kind of, I think that was happened prior to this, but that might have been, in my mind at least, that was one of the first events that kind of led to this outcome. If it's accumulating in people, then it kind of led to more scrutiny in this. Sure. I'm almost done. So a little bit about like biosolids and kind of where PFAS is, right? So on the left there is just kind of a graphic illustrating soil. It's a porous material. There's pore space where water is present, and on the right is a diagram that just shows sampling of different biosolids, some that are non-biosolid based and some that are biosolid based soil amendments, and then the different types of PFAS compounds that were found in them. So really all this is meant to show is that the mobility of these compounds in soil is really a complex function of the soil composition, the chemical structure of the molecule, the water quality. There's not a really direct, simple way to understand that transport and fate. And similarly, the presence and composition of it in soil amendments like biosolids is variable, right? You can see from this list, the composition in one is quite different than the other, right? So there's not a straightforward linkage. Yeah?
So it says presence and composition of PFAS and soil amendments is highly variable.
Mm-hmm.
And since you're working with such drastically small quantities like parts per trillion, could theoretically a load of biosolids, like one shovelful or 1 teaspoon, test hot and the rest of it not, or is it homogeneous across the board?
Oh, I see. Yeah, how homogeneous is it? That's a good question. I'm not sure, but I would guess from what I've learned of how it's made, it goes through so many different oxidation steps, inactivation steps. There's so many steps in which it's mixed and homogenized that I would assume that to be the case. But that's actually a good question. I don't know whether there is inhomogeneities that could occur. I mean, it's plausible that could happen. Yeah.
I just had an experience at a farm that we would work at that had biosolids spread over several years. Last was in 2018, and I tested soil and the surface water that was there, the wells and everything. They all came back negative and so small, undetectable. The only one that was high was where they dumped the biosolids to then spread it.
Mm-hmm.
But it was so undetectable as well. And most of the soil around here is clay. And because it's so mobile, I wonder when they're doing surface applications like that for sod, grass, that it's running off or it's getting cut off from the grass and moving out of spite of that. Because the level's so small now that it's not a problem.
Yeah. Yeah, I mean, I guess if you don't see it there, it doesn't mean that it wasn't transported elsewhere. Yeah. That's the challenge of detection, right, and kind of being able to trace these things. So this is a little bit of just where the current regulations stand. So there's guidance documents from the EPA, but not actual regulations. So it's kind of a state by state basis. So Maine is the only state that has a land application ban, and they have a ban on the sale of biosolids from wastewater treatment plants. Connecticut has banned the sale of biosolids that contain PFAS. Maryland has an active monitoring program. New Hampshire and Massachusetts require sampling. And then there's kind of a group of states that have various kind of levels of development. So that's kind of where things currently stand. And there's different tiers that kind of classify what the intervention is. It can be as simple as land application allowed. So if it's below 20 micrograms per kilogram, that's kind of consistent across these different states versus tier 4, which would be prohibited for use. So depends on the state, depends on how each state wants to approach it, but that kind of gives you a snapshot as to what different states are doing right now.
Do you know what Maine is doing with their sludge, since they're not applying it to land? Are they doing like Maryland, which is to send it to Virginia?
Yeah, there's an article, if you read recently, saying there's sort of a capacity glut, and they're kind of struggling at the moment to figure out what to do with it. And that's a different type of challenge, right? Is what do you do with this material? So that was a recent article I was reading. So they're kind of projecting a there's like a projected catastrophe, they said, because they're going to have all this material and not know what to do with it. So that's where I think people are doing research, like Mike and myself, to figure out ways of trying to remediate this stuff. I'll share this later. This is just from the EPA's website. So these are, if you're concerned about biosolids applied to your farm, these are sort of recommendations the EPA gives you. It's the one at the bottom I found humorous because it said, learn more about what's happening in Maine. So Maine's a state that's been addressing this. So Maine is really a place that's kind of been leading in a lot of ways. But there's links here to local, state, federal, links to do testing, all these different things. So that's kind of guidance that's given from the EPA. And this is one study. This is kind of a more quantitative study. This is from the Maine biosolids studies. This is a retrospective study over 3 years, 145 participants. Over 70% of them lived within a mile of land where biosolids was applied for agricultural use and well water testing. Their well water was tested. About half of them had well water which exceeded the Maine drinking limits. And so this graph is kind of showing the serum concentrations. So just a reminder that y-axis is 1 part per billion. So serum levels are elevated and highly correlated with well water concentrations and biosolids applications. So if you go to the CDC's website, the average serum concentration, based on studies, is somewhere in the 2 parts per billion range, or 4 parts per billion, depending on the specific molecule. So, and this was up around 10. So there's another study that kind of links the application of biosolids and the exposure of it, in some cases through well water and drinking water, and other cases through other routes to accumulation. And just to wrap up, then I want to talk about some of the work that Mike and I have been doing, to kind of start to address some of these issues. So we work with growers in Maine. So this is Aroostook County. This is the northernmost part of Maine. Randy is in the picture here. He's the director of the Soil and Water Conservation District. He's a farmer. He also teaches at University of Maine. And so, that's his farm. We're having a conversation there. Like in true Maine, it's like 40 degrees out there, but he's in shorts and a t-shirt. So, and then on the right is, broccoli and Brussels sprouts, and you'll see he labeled them PFAS, broccoli and Brussels sprouts. So, he had been testing irrigation water from the Aroostook River. That contained PFAS, and so he was tracking that through the crops that he was growing. So Mike and I are working with Randy and other growers up there to assess the spread of PFAS in soils and crops during a typical grow season. And so this is a report from 2024. This is a study that we've been doing with Randy. So potatoes are a major crop in Maine, in northern Maine specifically, so the red Norland specifically. So we sampled the edible and inedible parts of the potato, as well as the irrigation water sources and soil. And interestingly, what we found was that in the edible part of the potato, we did not detect any PFAS compounds, but in the vegetative parts or the leaves, we did. And so, as Mike pointed out to me, that's where most of the water is present in the plant, and therefore that's likely why it's accumulating there. So in one sense, it's good, right? So if you're a grower and you have a baseline contamination level, you can still grow your potato with decent risk mitigation to know that your crop is safe. But at the same time, you don't want to plow that vegetative tissue back into the ground, right, and recycle it back in. And so we're in year 3 of this now. We haven't finished collecting this data for this year, but we're trying to collect this year on year to start understanding how this happens year in and year out. Yeah.
Do you know how it was irrigated? Was it drip or was it overhead irrigation?
Drip, I believe.
Okay.
But I'd have to check. I don't remember. And that's just one example of the kind of things that Mike and I are doing both in the lab here at UVA, in kind of a laboratory setting, and then out in the field to try and give practical recommendations to growers. The other thing we've been working on is trying to develop these kind of low-cost biosensors for field use, right? So if you were to do a test at a commercial lab, it's anywhere from $500 to $1,000 to do the test. There's about a 10 to 14 day turnaround, so it's pretty expensive. And if you're a grower, that's probably a prohibitively expensive test, especially if you want to sample periodically throughout a grow season. So Mike and I have been working on developing these sensors that are kind of low cost, kind of point of use sensors. So they're not going to be as precise as what you would get from that other test, but it's something that's direct, effective, reliable, fast, and relatively cheap. So it's kind of a quick yes or no. Is kind of how we've approached it. So there's that star is kind of where Loring Air Force Base is. We're all the way up on the north easternmost corner of the U.S. That's where Loring Air Force Base is. So that's land that now belongs to the Micmac Nation. There's kind of a history of waste oil, fuel, solvents, PCBs, firefighting foam, and other things present in ground, surface water, soil, and sediment. So it's a pretty challenging environment. So that's where we had begun to do some of the testing. And so we published a paper last year where we sampled surface water sites. This is a map where you can see the Loring Air Force Base. You can see the Aroostook Band of Micmacs Trust land adjacent to it, and then some of the water from Chapman Pit and Malabeam Lake. And we sampled those. We did independent testing, then we compared the results to the sensor that we developed. And so you can see that the sensor as developed is pretty biased towards finding PFOA, which is one of these two major compounds. So that's why in Chapman Pit, which is pretty much abundant in PFOA, we see a good agreement. In Malabeam Lake, PFH excess was a Gen X, short chain PFAS was present at a high level, so we didn't get quite as good agreement, but it's still correlative. And so, at least at a first pass, this was a validation of these things being used in an environment. We could do these tests in the lab with just a basic fluorimeter. And so now Mike and I are working on trying to refine this, improve it, and then get it to a point where it could be more kind of field deployable. And then you'd asked to mention the hemp work. So in Mike's lab, we've been evaluating different plant types. Mike can speak more to that than me. And looking at how different plants can take contaminants of concern up, like heavy metals or PFAS. So industrial hemp is one that we have been studying. There's some old data kind of showing what I would call like sorption capacity for these different compounds. So micrograms per plant. So I showed you before, like in the blood, that was 10 micrograms found. So that's kind of the same scale. So the plant has a pretty high sorption capacity. And we're hoping, maybe this summer or next summer to begin to evaluate how these could be used in rotation or in combination in a grow cycle, or other plants that Mike has identified that might help mitigate this.
So then these remediation plants, when used, are they just sent to landfill?
What do you do with it question.
You're saying to not turn them under.
Right. Yeah, that's sort of the challenge, right? Is that what you can sequester it, which is and it's kind of like with the potatoes, right? Being able to give growers recommendations to protect the crops, but you're still left with the challenge of breaking it down. Mike and I and a lot of other groups too are working on ways of destroying these molecules. That's a much more challenging problem because, as I mentioned, it's the strongest bond in organic chemistry, and there's no known terrestrial process that uses fluorine. So finding that solution is challenging. There's incineration, there's other techniques like this, but they're only partially effective. So that's still, unfortunately, a challenge without a good solution. Yeah.
This is a biochar is a kind of niche place that vegetative matter could get sent to. Do you know if PFAS survive the process of the creation of biochar?
Yeah, that's something we're actually actively working on right now with USDA. Mike and I have a project where we're evaluating hemp, and that's taken PFAS up and seeing whether we can break it down. There's a lot of other published studies that suggest there are different ways in which it can decompose during the biocharring, like pyrolysis process. So I don't know, Mike, it's kind of an open question, but there are studies that show it's possible to at least partially break it down.
Possibility that if you heat it high enough, you can get it to break down.
Yeah, because the solids part absorbs the heat, right? The carbon containing part. So that heat which is taken away in transforming the material is heat that would have otherwise gone to the breakdown of the the PFAS molecule, right? So there's kind of this intricate balance. Should it be hydrated? Should it be dried? There's a lot of these kind of variables that we're exploring, and others, you know. So I think it's an open question. Good. And then, yeah, I'll just mention another way in which we're looking at this is, I launched this company a few years ago, and we're trying to develop, so I mentioned pesticides are a major source of PFAS. We're trying to develop biological replacements to pesticides, so just completely eliminating these chemicals entirely and replacing them with something biological. And so we're working on, so fire blight, if there's any apple farmers here, or apple growers, that's a major disease that's affecting crops here in Virginia as well as out on the West Coast. It's a bacterial disease that it's called fire blight because it kind of the trees look scorched or burned when they get the infection. Currently, antibiotics are sprayed on the tree as a way to try and mitigate the disease, and that can lead to environmental antimicrobial resistance. These are some pictures of what it looks like. So if you're going through an orchard, you see that ooze coming out of the base of the tree. That's the pathogen kind of emerging out of the tree and overwintering. That's the ooze on pear surface. That's what it looks like. And so, we'd like to try and eliminate or reduce antibiotic use and find a better solution. So this is actually we work with colleagues in Oregon and Washington as well. Washington far and away is the largest apple producing state. They dwarf the rest of the United States. Yeah.
Are there particular classes of pesticides with higher concentrations of
I couldn't hear the second half of the question.
Specific classes of pesticides that have higher concentrations?
Classes of pesticides that are at higher concentrations?
PFAS.
That's another open question. That's actually something Mike and I are hoping to study as part of this grant. We kind of proposed to try and test that. If you go to the EPA's website, they did some sampling and testing, and there they found it was fluorination of the packaging. Because remember I mentioned earlier, like fluorination of the packaging can generate PFAS as a byproduct. That kind of prevents clumping or granulation of it. So if you have it sitting in a barn all summer, you don't want like a big brick when you take it out. That's why they fluorinate it, to prevent that from happening. So it's a little dependent on the specific batch or lot that was made based on that. That was sort of the outcomes from that EPA website. And so we work with colleagues at Virginia Tech. So Surgeon's an extension agent that specializes in fruit trees. And so this is one result. So we have a USDA grant that's kind of supporting this work, where our experimental product, which is CAACE, is about as effective as Firewall, which is the antibiotic which is currently used, and approaching similar efficacy to Firewall or Fireline, which are this kind of antibiotic combination. And so we're trying to either combine it to reduce antibiotic use, or maybe in some cases get rid of antibiotic use entirely. And so a surgeon and colleagues at Oregon State and Washington State, we're hoping to try and mitigate this in a different way. So that's all I've got. So I'm happy to answer other questions, and thanks for your attention. Yeah, you got it. Yeah.
There are just some things, some questions I want to put to Bryan, because the industry and DEQ, which has been so adamant that we do continue to use biosolids, has some certain predictable lines of argument. So one of them is, well, you've been working on soil that's been contaminated from an Air Force base. So what does that have to do with biosolids?
Yeah, so how would I answer that question?
Yes, how would you answer that question?
I guess, I get this is kind of like the fingerprinting question, right? Did it come from, because last night at the webinar someone asked that question, like what if it came from rainwater? What if it came from here? How do you know that it ended up due to biosolids? I think the reality is, like, I go back to the data that I showed before. There's routine testing that show biosolids, contain PFAS, and so if you're sampling routinely and you know it to be free or clean of PFAS, I guess it's safe. But from all the testing we've done, we see that that's really not the case. And so, trying to say that there are other things that are worse than it. I don't really agree with, because if you look at the bioaccumulative potential of it, like the animals being an example, right? It's parts per billion versus parts per trillion, right? No matter how you're introducing it into the soil, it's going to end up in, the food we eat and us. So anywhere we apply it to agriculture is going to be bad for us, no matter where it came from.
And there are a few other things besides PFAS in biosolids, right? Like, what are some other things that accumulate?
How about applying it to like lumber or like tree growth? I don't know.
Yeah, I'm curious.
If it accumulates in wood, would it get locked up into trees?
Oh, the PFAS compounds, you mean? Yeah, we're looking at the northern ash tree as part of the grant, because that's actually that's important in basket weaving, which is a tradition for the Micmac Nation, and so we're actually sampling that. I'll know by December what we found, so that's something that we're trying to test now. Yeah.
Are you saying you're trying to see if it's in the tree rings, or like what do you mean by if it's in the trees?
We're sampling it's the bark and then kind of the outermost parts of the tree, because that's the parts that they use in their kind of traditional basket weaving. And there's actually a lot of barrel making in Maine as well too, from ash and other wood, and so we're sampling some of those barrels and other things to see whether there's leaching out of those barrels. So.
Yeah, so for those who don't know, it's not just farming that biosolids are spread on, it's silviculture as well. So it's been, you get cut over woodlands and then they'll come in and spread the biosolids before they plant.
Yeah, agroforestry, there's all sorts of other ways in which it's applied to. Yeah. Yeah, and your question, that's not there's oils, fuels, solvents, cleaners, PCBs, heavy metals. These are all found in biosolids as well too. So it's not only PFAS. It's just like you said at the beginning, that's of particular concern and kind of raised specific attention.
Yeah, so, go ahead, if you have more questions.
So I'm one of the map of one of the houses that's right next to the one that's not too far away from here that had biosolids spread not too long ago. And so we have it well tested when we moved there, but as I'm sure you know, there's only like 2 labs or something in the whole country where you can get your water tested for PFAS. Are you guys looking into anything to make that more accessible?
Oh yeah, yeah. I mean, Mike and I talk about this all the time. I mean, we're hoping. I mean, there are different ways in which we've been trying to. The idea would be to set up a kind of a testing lab, maybe affiliate, like in Maine, for example, University of Maine has a testing lab, and we work with another independent testing lab. If it's a university-associated one, it could be, for example, we're a public institution, more of a community-based or kind of subsidized testing model. Maine has gone to that. I don't know if other states have yet or not, but that's something that, we've certainly talked to folks in the state about.
Because it wasn't that it was really expensive, it was just there was not a lot of options. And
right.
I think like one of the labs was trying to start a new lab, and so they weren't even taking new samples for like 6 months or something.
Right.
It was just like you couldn't get your water tested even if you wanted to. And I think the test was like $99. It wasn't like $1,000 or something.
Yep.
But it was just there was no capacity.
Yeah, I would say too to that, there's a little bit of like buyer beware as you look at some of these labs that are starting up. This last night that came up in our, I don't know if that was in the after conversation. There's I was on a panel last night where one of the speakers specializes in setting up testing labs and consults for these groups. A lot of groups that might do testing, say in like testing for pesticide residue, say in agricultural products, are now switching to PFAS, but it's not the same type of testing necessary. There's unfortunately a lot more cost and sophistication. So, not to say it's bad, just that sometimes I would look to see whether it's the EPA 1633 test that's done, because that's the actual EPA standard. If it's not that, then you're running the risk that maybe it's not, what you're hoping. So, not to say that it isn't true. I just don't know in this case. Yep.
So, and going on that as well, what's the, from the outer world, bringing it back to me,
Yeah, I mean, I don't think, Mike and I work with, it's Maine Testing Labs. So that's a facility that Katie Richards started in Maine, and she's a methods development person. So she's developing methods for soil testing, food testing. So there are places like that, but it's more kind of experimental or developmental. I think the EPA only has a current standard, I think, for drinking water, the 1633. I think there's a 537.
It's not promulgated, but it's approved for soil and biosolids
Right.
as well.
Right.
So it's sort of like there's one step shy of it being absolutely 100% approved. But if you're a farmer, you can get your land tested for PFAS and get reimbursed for the cost of the test. If you know how to navigate the NRCS office, and it's USDA money for it. So you go and sign up for they call it an EQIP program. It's sort of in the same categories like riparian protection programs and things like that, where you plant trees and they get reimbursed. And it's called the CEMA 209, CEMA 209. I called our local office and he checked, and the money's still there, hasn't been taken away. But nobody's using it because they don't want to know, because there's no safety net. What Maine has done while they're leading is they've created a safety net right off the bat. They did it very quickly, and they put the Organic Farming Association in Maine in charge of dispersing it. But it's to help farmers directly, like they'll buy your farm, they'll help you relocate, they'll help you with whatever you need if your livelihood's been destroyed by this. And so farmers know, okay, I can test and find out, and then I'm not just left up hanging out to dry. And then there's money to help with health problems, physical and mental, starts next year. I guess they ran out of money, so mental health is waiting. And then, what is it, land, health, what's the other thing they're doing, Brian? I'm forgetting. It's research.
Yeah, yep, research, yep.
So funding research for, specifically to help farmers, the mitigation of their lands and crops and so. So that's why you have partners in Maine, right, in there. So a lot of that money has gone to University of Maine or other, institutions and organizations in Maine. So that research money actually becomes an economic driver, which is kind of cool. It would be nice if Virginia did something like that. I wanted to address the solutions problem. That's why I started talking about this, because it's sort of a short-term and long-term answer, right? So short-term, you have to stop. You have to stop spreading it, and then landfill it. But in order to do that right, you have to de-water it. So that would need to be a requirement. And then you can have this dry cake that actually is an excellent day cover over landfills, and it won't take nearly as much room up as like throwing the wet stuff on landfills. And I think, you can't do it forever, and that's kind of the point. It puts some pressure on, right, to get the long-term source control, stop making the stuff, product bans, that kind of thing. Get more money into research. So, and because a lot of companies are seeing the writing on the wall and really trying to take advantage of this and coming up with some really cool options for at least, they're starting with cleaning water because that's the easiest thing before you try to cleaning sludge. But there are, just today I learned Denmark banned PFAS in clothing and footwear. In Germany, they've got up to 85% of sewage sludge is being remineralized. That means they're heating it up to break the bonds, right? Through fluidized bed combustion. So if you heat it up high enough, it will break the bonds. It's just an unfortunately energy-intensive way to go. But there are some low-temperature things coming along. Chemical destruction that produces just plain old fluoride at the end. And running electrical current through it.
Electrolysis.
Yes. That's another promising one that is not as energy intensive. So all hope is not lost. We just have to, look this squarely in the eye and do something about it and not just keep putting our heads in the sand.
I think reducing the amount of products we buy that might have it, clothes, shoes, cleaning products.
Yeah, it's just that it's so ubiquitous, it's really hard to avoid.
Can you briefly describe how the testing works? Is it like electrospectrometry or?
Yeah, I think the EPA methods are LC, mass spec, so it's LC-MS-MS. So that's it's a pretty expensive equipment, and that's kind of the reason why the tests end up being expensive. You need sort of a trained scientist to run it, so it's not a straightforward thing to set up. And then the cleanliness of the space, because it's in air, it's in dust, so your background signal can be high. So that's kind of the current test. That's how it's done currently.
And how's your sensor different from that?
Yeah, ours is just a direct application. So it's a protein that binds the molecule, and when it binds it, it changes color, and so that color is the indication that it's present. So it's like we're working with a company now that makes like lateral flow tests. So like your COVID test, when you do the swab and you put it on, and if the color changes there, it indicates the antigen, so you've got COVID. It'd be a similar type of format like that, ultimately.
You have a protein that specifically binds to only PFAS?
Yeah, so in this case, PFOA, that's the primary one that it binds, and we're kind of working on ways of changing it to maybe bind others as well. So right now, PFOA to start with, and then hopefully others in the future.
So it's a little after 7. If y'all want to hang longer and grab a snack, you can do that, and we'll Dr. Berger's going to hang
Yeah,
on
Mike
a little
too.
bit.
Yeah,
And
Mike will be
too,
here as
so
well.
We can attack them as needed.
Yep.
And you,
Yeah, thanks.